Hello everyone. Today I'm glad to share one of my interested research area about the COVID-19 infection in the world. I'm Zhong Mei and come from Jeffrey Chin School of Medicine and Health Science. Maybe the possible transmission of COVID-19 have many different routes. But it's well known that severe acute respiratory SARS-CoV-2 causes COVID-19 pandemic and mainly triggers acute respiratory distress syndrome and viral sepsis and brain challenges to the patient's treatment. The pandemic infection of COVID-19 confirmed nearly 16 million human cases to now, including more than 6 million deaths, and the infection rate is continuously increasing worldwide. The present outbreak of COVID-19 has been labeled as a global pandemic by WHO and has posed critical challenges for public health, research, and medical communities. Airborne transmission has been recently reported as a dominant route for COVID-19 infection. Similarly, many research groups had reported gastrointestinal manifestation of COVID-19, proposing the facial oral route as an alternative route of SARS-CoV-2 transmission. This finding suggests that the epithelial cells of the oral cavity provide the binding site to enter and propagate the SARS-CoV-2. However, the expression of possible receptors of the SARS-CoV-2 in different cell types of the oral cavity hasn't been investigated yet. From this picture, we can know that SARS-CoV-2 entering into the cell is induced by the binding of viral spike protein to host cellular ACE2 receptor. Previous findings indicated that ACE2 plays an essential role in entering in the host cells. Thus, ACE2 expressing cells may act as target cells and are susceptible to COVID-19 infection. Some scholars have reported the ACE2 receptor expression in various human tissues, including the epithelium of the lung and the small intestines, as a co-expressed membrane endopeptidase of the ACE2 receptor. Furin has the potential role to cleave the viral envelope glycoprotein, thereby enhancing the viral fusion with host cell membranes. Genomic characterization of SARS-CoV-2 has revealed that the furin enzyme could activate the specific site of its spike protein. From this picture, we can know that presence of viral entering receptors on the oral epithelium may lead to some possible outcomes of oral manifestations, such as ulcerations, blistering lesions, loss of taste, and even systemic spread. But in the world, some scholars are interested in do some public single cell analysis, like the picture shows that single cell RNA sequencing technique provides an avenue to understand gene regulation networks and the complexity of cell-to-cell heterogeneity at the single-cell level. Single-cell RNA sequencing has diverse applications on bioinformatics, stem cell differentiation, organ development, whole tissue subtyping, and tumor biology. Unlike single-cell sequencing database analysis, unveiled that furin and ACE2 are expressed in potential target organs such as lung, heart, nose, rectum, colon, intestine, and ileum. Although the expression and distribution of ACE2 and furin in the epithelium of the oral cavity could be critical for the COVID-19 invasion of the host cells, but the information on the expression and distribution of them in the human oral cavity is limited. So we also acquired the public single-cell RNA sequencing database and do the sequencing analysis. At the same time, we also collected 12 normal tissues of the oral mucosa, included 3 buccal and gingival tissues, 2 lip, tongue, and palatal tissues, respectively, were taken from 12 different patients with clinical diagnosed fibrous epithelial polyp or benign tumor, whose average age was 53 years. When we got this species, we performed immunohistochemistry analysis for ACE2 and furin in human oral epithelium tissue sections. So the results are like the following atoms. The first one, after analysis single-cell database, we acquired the result about the identification of 16 cell types in oral mucosa tissues, including epithelial cells, fibroblasts, T cells, and B cells. The heat map, the right picture shows the heat map of the main corresponding cell marker gene expression profiles across the cell types is plotted in. The marker gene of epithelial cells included KRT6B, GJB2, SFN, and KRT14, like this. The next result is that ACE2 and furin expression in the oral mucosa. The volume and the scatter plot showed the expression profile of them. ACE2 and furin are mainly expressed in epithelial cells. The percentage of them positive cells in the oral mucosa. So at first we have acquired the result about the expression of ACE2 and furin in the database. So next step we want to use the IHC to check whether they are expressed on these tissues. From this picture, we can know that ACE2 protein is highly expressed in the epithelial layer of the buccal mucosa, lip, palate, tongue, and gingival. Maybe they have some difference about the expression. Next picture is a furin protein is highly expressed in the normal oral mucosa. According to the results about IHC tests, we concluded this picture. Maybe they have some difference about the expression. Like the furin is strongly positive in lip tissue. ACE2 positive in buccal, lip, tongue tissue, and furin positive in gingival and tongue. ACE2 is weakly positive in gingival and plate. Furin is weakly positive in buccal and plate. So these are the all results about this research. So according to this, these steps to acquire the expression and study the transmission via the respiratory route. Maybe we have some discussions. The first one is 2K proteins, furin and ACE2, may play an important role in entering the target cells in different routes according to the oral mucosa, which implied the potential attack and spread of SARS-CoV-2 in the oral tissues. It is suggested that all healthcare workers should focus on taking effective measures to prevent the virus from spreading via the oral cavity, and the potential impairment of oral tissues should be evaluated after infection with SARS-CoV-2, giving more evidence to the prevention and the treatment of COVID-19. Strict precautions should be taken to protect from direct exposure of oral tissues to SARS-CoV-2 contamination. Our results would be valuable for the prevention and management of the COVID-19. So, in the end, in this special period, we must protect ourselves and ensure safety, especially like the doctors and some some like this. Yeah. So this is today's sharing. Thank you.